Hi, back again, Dr. Bones Show. We were talking about foods the other show, and foods provide energy, and vitamins, minerals, fiber, phytochemicals, meaning plant chemicals. And we talked about muscles needing energy and so on. So of course you need energy for muscles, but certain forms of energy are better than others. For example, if we imagine that this is a protein molecule and each of these individual toys are the amino acids, well, we could use proteins for energy, but it really wouldn't be a good idea, right? You'd want to use proteins and their amino acids for the building blocks for proteins in the body, meaning muscle tissue. So not a good idea for energy. But there certainly are other forms. We talked about glucose, all right? So this is the linear form for glucose. It's got 6 carbons, 6 oxygens, and 12 hydrogens. If we look over here, we see a molecule that we use regularly in the morning when you put some spoonfuls in your tea or coffee. If you go to this slide here, this is called sucrose, all right? So sucrose is a glucose unit and a fructose unit, all right? So this would be something you would get from grapes, grape sugar. And of course, fructose is really basically the same thing, just sugar that you would get from different fruits and vegetables. But they call this grape sugar, all right? So the sucrose molecule or table sugar could be useful for energy. And if you needed a long-term supply of energy, you would need fatty acids, all right? So fatty acid molecules come in fats, triglycerides. The long-chain fatty acids, there are 3 of them, 1, 2, 3, that hang on a molecule called glycerol with 3 carbons. So a 3-carbon glycerol, which can also be used for energy, and 3 fatty acids, all right? Some of these fatty acid chains can have as many as 24 carbons, so that's quite enormous, an enormous supply of energy, all right? So that's the way you should get your energy, from sugars and carbohydrates and fats, all right? So fats and carbohydrates. Now, if we wanted to use energy in the body, and if you can zoom in here, we're going to talk about something called glycolysis. So if you wanted to zoom in here on glycolysis, we see that glycolysis, which is in the cell cytosol, we're going to convert those sugars, in this case glucose, which has the 6 carbons, into pyruvate, which has 3 carbons. So you're going to take the 6-carbon glucose and basically cut it in half. So you end up with 2 pyruvate molecules, right? And the pyruvate can then enter into something called the Krebs cycle, which is also known as the citric acid cycle, and then those 3 carbons can be worked on for more energy. And the energy in this case, if we pan over here, for cells is called ATP, adenosine triphosphate, all right? It's little parcels of energy. So if you took a glucose molecule here, which we have one over here, if you take a look, all right, down here, all right, we see the 6 carbons, the 6 oxygens, a lot of hydrogens, all right? And we were to use that directly, that would just be too much energy for the cell. The cell needs little portions of energy, all right? So that will come in the form of adenosine triphosphate, with the phosphate bonds being broken and put together, all right? So we'll be talking about that as we go. It's a wonderful way for cells to get energy. Sort of like when you go somewhere and you've got dollar bills and you want to use coins, right? So you want to put coins into a machine. You don't necessarily need to put dollar bills, although when I was a kid, I guess you could put pennies in there and get a gumball. Now maybe you have to put in $1 to get a gumball. I don't know anymore, but in any case, probably I shouldn't be eating too many gumballs. All right, so you put in your pennies, all right? So ATP is sort of like coins, all right? Instead of dollar bills being the glucose molecule, much too much energy, ATP, a small parcel of energy, all right? So the cells can use them. Very useful. All right, so in the next segment, I'll be coming back to talk about the various steps in glycolysis. There are 10 steps, and we will go over each of them, and we'll talk about the enzymes that are involved with glycolysis. So I'll see you in the next segment. Take care. Bye for now. Hi, back again, Dr. Bones. We're talking about glycolysis. I mentioned that glycolysis is, in essence, cutting glucose into pieces, all right? In this case, we're going to cut glucose into 2 pieces. Eventually, the glucose will form pyruvate, all right? So going from the 6-carbon glucose to the 3-carbon pyruvate. Here we see a glucose molecule, all right, the 6 carbons, has 6 oxygens, 12 hydrogens. Well, when we use carbohydrates for energy, we're trying to form ATP. We're adding a phosphate group to adenosine diphosphate to create the adenosine triphosphate. So it's a wonderful change machine, all right? You can add coins and subtract coins and so on. So the cell needs certain small parcels of energy. So it's a wonderful way for cells to get energy. All right, so 10 steps in the formation of the conversion of glucose to pyruvate. There is a preparatory phase where you have to add some money, in this case 2 ATP input, all right? Sort of, you want to start a business, you got to have a little money to put into it for the payoff, hopefully. Hopefully you don't go out of business before the payoff. I would hope not to go out of business before the payoff, all right. So in any case, so we're going to put in 2 ATP, all right? So we're going to show you how this is done. In the payoff phase, there's a generation of 4 ATP, all right? So 4 ATP, if you can zoom back in there, so 4 ATP output and 2 NADH. These are basically energy units that you're going to use, all right? So energy in, energy out. Of course, all of the energy is coming from glucose. The glucose you eat, converting to pyruvate. So without this, there is no energy. But we want it in the form of ATP. All right, so let's go to the first step. Taking glucose, if we go over here to this board, we've got sucrose, all right, which we have glucose and fructose, all right, so this would be the glucose. And again, you can take the glucose and make a linear structure, all right, so in any case, it doesn't matter whether we have the cyclical structure or the linear structure, we have 6 carbons on board. So we're going to be breaking those bonds eventually to form new bonds, but bring to the forefront ATP. So with the glucose molecule in mind, and here I am, here I've got my glucose, so yes, you can actually go to a hardware store and take these fold-out rulers and form molecules. So this is kind of neat. You've got the hexagon-shaped glucose molecule, right? I haven't put on all of the OHs and hydrogens, right? There's be too much going on, but you see the 6 carbons and the oxygen. All right. So the first step is to add phosphate. So you're going to add phosphate from an ATP molecule. So here we go. Take a look here. Step 1, we start with glucose. So here's your glucose. And we're going to add a phosphate from ATP to form glucose 6-phosphate. Now if we go over to this board here, we can see the numbers. As we go back and forth, it's easier to just take a look. Here's carbon 1, 2, 3, 4, 5, 6. Well, the same thing over here, all right? We've got the 6 carbons. So this is carbon number 6. We're going to add a phosphate. Phosphate, if we go back to this board, is this molecule, P for phosphorus, and 4 oxygens surrounding it. We are going to add this phosphate to this carbon, right? Now, on my ruler structure, I'm not going to add the PO4, I'm just simply going to put on a phosphate in the form of the letter P to represent PO4. So there we go. This is called glucose 6-phosphate, all right? Here's carbon 6, here's the phosphate, and now it's attached to the glucose molecule. So the first step in glycolysis, an ATP was used to put a phosphate on to glucose. So glucose 6-phosphate. All right, now let's go to the next step. Here we see on the board, we've got Step 2, fructose 6-phosphate and a water molecule here. Yes, a water molecule was used or is used in a rearrangement. Actually, before we go to step 2, let me go back to the enzymes that are involved. When we went to step 1, we talked about moving a phosphate onto glucose. Well, that will take an enzyme. All right, so if we look at step 1 here, we see the glycolysis enzymes. So glycolysis enzymes, the first step, needed hexokinase. All right, that is called a transferase. It's going to transfer a phosphate onto the glucose. So it went from glucose to glucose 6-phosphate. It needed the enzyme hexokinase. In the next step, we will see the next enzyme. So there is a rearrangement done, and if we go back over to this board here, you can see what I'm going to be doing with this molecule. Go back over to the board. So here would be the glucose, right? And we need to rearrange the molecule into fructose. So this phosphate on glucose is now going to be over here on fructose when we create fructose 6-phosphate. Again, glucose is here, fructose is here for table sugar. We're working with the individual sugars now. Either we're working with glucose 6-phosphate, or we're working with fructose when we do the rearrangement, fructose 6-phosphate. All right? So I'll be back with this rearranged molecule in this form, right? Glucose 6-phosphate. I need to rearrange it into fructose 6-phosphate. So I'll see you in the next segment.